My time hasn't started yet, has it? No.
This slide should be top.
Hi, my name is Minyi Lee, and this summer I worked with Dr. Erin Golden in the genetics department of the Albert Einstein College of Medicine, analyzing the genetic effect of low Earth orbit on mice. So 2 previous independent experiments sent mice to space, and then upon arrival extracted the RNA from the thymus and the skeletal muscle to determine which genes were upregulated or downregulated in low Earth orbit, meaning which genes were more expressed or less expressed. And so what we wanted to do is take the raw data from those experiments and reanalyze it to see if there's anything in common. So just some background, low Earth orbit, or LEO, is any orbit that's between 160 kilometers and 2,000 kilometers. It's the location of many satellites, and it puts an organism under the unique stresses of weightlessness and high radiation. A microarray, which is what was used in the experiments, is a method to measure gene expression, and it takes advantage of the fact of the natural chemical attraction between DNA and RNA molecules. So DNA has 4 bases, C, T, A, and G, and RNA has the same 4 bases except instead of T, there's U. So C is complementary to G, meaning that C binds with G, and A is complementary to U. So if you have a single strand of DNA and a single strand of RNA and the bases are complementary, then the 2 strands will stick together. On the Affymetrix chip using the previous experiments, there are about 500,000 cells, and in each cell there are millions of strands of DNA. And using this, so before the experiment, you take the RNA, copy it a bunch of times, and tag it with a fluorescent compound, then wash it over the chip, and during which the sample and the DNA strands on the chip hybridize or stick together. Then you shine a laser on the chip, which makes the fluorescent compound glow. And so if there's more hybridization, which means the gene is more expressed, then it'll be brighter. Then you use a computer to compare a chip from the control experiment and then the sample to see if what the change in gene expression is. So these are the 2 experiments that we analyzed, and so they sent mice to space for about 11 to 13 days. And extracted RNA from the thymus and the muscle. And the reason they chose these 2 tissues is because the thymus is related to immune function, and a few of the main concerns when you go to space is loss of immune function and muscle loss. So what we first did was reconfirm the results of the paper, and we got the raw data from Gene Expression Omnibus, which is a website, which is just a database, and using a branch of that website called GU2R, we determined how much the gene expression changed and then confirmed the results. And we intended to see if there were certain genes that were upregulated or downregulated in both experiments, but we couldn't really find that. And afterwards, we tried this program called Ingenuity Pathway Analysis, which tries to draw relationships between the inputted genes, but we couldn't really find anything conclusive. So lastly, instead of focusing on the genes that are most upregulated or most downregulated, we wanted to take the summed effect of all the genes and which pathways they relate to. And to do this, we used an R package, which is a programming language called MetaPath. And we were expecting to find pathways related to DNA repair, apoptosis, or stress response. So these is an excerpt of the results that we found from MetaPath. So the left-hand column is the pathways, and the right-hand column are the p-values, which is a statistical value for how significant it is. And so the 3 highlighted pathways are ones that we picked out as interesting because they were what we expected to find. So the first one is related to a response in UV radiation. The second is a response to stress on a genetic level, and the third is associated with many human diseases, such as cancer, which makes sense because there's radiation. And so in the future, we wanted to expand our study to do the same sort of analysis in bacteria and see if we find similar results. And instead of using MetaPath, we wanted to use a different program, but we couldn't get that program to work while I was there during the summer, so that's ongoing. I would like to thank Dr. Aaron Golden and Dr. David Reed for mentoring me, Dr. Cox for putting this whole thing together. Thank you.